Hello everyone and welcome back. This is chapter 19 and we'll be discussing disorders of the immune system. The types of immune system disorders that you should understand by the end of this chapter are hypersensitivities, autoimmune diseases, transplant, rejection, and immunodeficiencies. The first one, hypersensitivity, is an immune response caused by exposure to an antigen that is undesirable. The antigen in this case is called an allergen. There are four types of hypersensitivities. Type 1 are anaphylactic reactions. Type 2 are cytotoxic reactions. Type 3 are immune complex reactions. And type 4 are delayed cell-mediated reactions. Type 1 hypersensitivity reactions, the anaphylactic reactions, occur when IgE antibodies bind to antigens, which recruits basophils and mast cells to release histamine. Histamine therefore causes swelling and redness, mucus secretion, smooth muscle contraction. It can occur within 2 to 30 minutes after exposure, and it may be systemic or local. So there are two different types of anaphylaxis. There's a systemic anaphylaxis and a local anaphylaxis. Systemic anaphylaxis can occur through different parts of the body. Blood vessels throughout the body can dilate and blood pressure can drop, leading to shock. Airway passages may narrow. A skin rash, nausea, and a tingling sensation may occur, and it also may be fatal within minutes. It's mostly treated with an injected dose of epinephrine to constrict blood vessels. Local anaphylaxis occurs in a certain area. For example, you can think of asthma as usually categorized as local anaphylaxis. When the antigens are respiratory allergens, they may cause itchy and teary eyes, nasal and respiratory congestion, and they are treated with antihistamine drugs usually. When the antigens are gastrointestinal allergens, they may cause an upset stomach or hives. So here are some fun anaphylactic allergen facts. First, 8 foods, milk, eggs, peanut, tree nuts, wheat, soy, fish, and shellfish are responsible for 97% of food allergies. Food allergy incidence has gone up. In children, it's increased 50% from 1997 to 2011. Nut allergy incidence in children has more than tripled between 1997 and 2008. And asthma incidence grew from 20 million in 2001 to 25 million in 2008. So many scientists are asking why. Why is there an increase in allergies? From which some scientists have created the hygiene hypothesis. And that is that our immune systems need normal microbiota and immune challenges in order to develop healthily. And there are several reasons why we might not be having this occur. This paper right here discusses Bifidobacterium infantis. It's considered a friendly bacterium in the lactic acid bacteria group, and it's been noticed to be missing in many people in developed nations. Since this bacterium aids in digestion, its absence has scientists concerned. The hygiene hypothesis would suggest that our clean or hygienic environments reduce the microbial challenges we get from different microbes, as well as exposure to beneficial microbes. That, in combination with the fact that we use antibiotics to treat other diseases, decreases the diversity of microbes that we have in our microbiota. Okay, so let's get back to hypersensitivities and let's discuss type 2 hypersensitivities, which are cytotoxic reactions. These occur when IgG or IgM antibodies bind to an antigenic cell and stimulate phagocytosis to destroy those cells. So an excellent example of this are cytotoxic reactions to blood cells after a blood transfusion. So individuals may be A-type, B-type, or O-type blood, and that concerns the type of markers that their red blood cells have on their outside surface. If a person in blood group A gets exposed to someone with blood group B, they will develop plasma antibodies or anti-blood group B antibodies and attack that, which will lead to a cytotoxic reaction. The same occurs if you have an individual that is a blood group B and they're exposed with A plasma or A red blood cells, they will develop anti-A. Sometimes people can have both blood group A and blood group B, and they will not form a reaction to A or B red blood cells as a consequence. Lastly, you can be blood group O, which, since you lack blood group A red blood cells or B, when you're exposed to that antigen, you'll develop antibodies towards it. So you'll have anti-A and anti-B antigens. And with all of these reactions, they take a certain amount of time for them to elicit a response. And this is typically seen in 5 to 8 hours after exposure. The most common type are transfusion reactions. So a little bit about transfusion reactions. In blood transfusions, only the blood cells are transfused, so the plasma containing the antibodies is kept behind. And as discussed, there can be markers on the outside of those red blood cells. Those markers or blood types are determined by the presence or absence of carbohydrate antigens on the red blood cells. So whatever your self type is, you will not develop antigens for that. If you're blood type A, you will never develop anti-A antibodies, but you can develop antibodies against non-self type antigens. So exposure of donor RBCs with X antigen, whatever it might be, to blood with anti-X antibodies results in what we call agglutination. And agglutination is just a fancy word for clumping of particles. This is also a concern when it comes to the Rh blood group system and a mother and her fetus. There's something called Rh factor, which people sometimes carry, sometimes they're positive, sometimes they're negative. If the father is positive, then the baby could be Rh positive as well. And so the fetus carrying those Rh positive blood antigens will circulate throughout the mother's body. And since that's a non-self antigen, the mother develops anti-Rh positive antibodies, and usually this isn't a problem for the first fetus, but if the woman becomes pregnant again with another Rh plus fetus, the woman will have a reaction to the fetus. The anti-Rh positive antibodies can cross the placenta and start destroying fetal red blood cells. And this can cause the newborn fetus to develop with jaundice and be severely anemic. So again, these are type 2 reactions. If we go back here, we see that these are type 2 cytotoxic reactions. Let's talk about the third reaction, which is type 3 immune complex reaction. So as the previous one largely had to do with reactions to cells, this one has to do with forming complexes. So it's caused usually by IgG, which is a free antibody that finds soluble antigens circulating in the serum, and a antibody antigen reaction occurs, and more of these occur, and it forms a larger complex that develops a deposit inside of an organ. This complex leads to the attraction of inflammatory cells to deposited complexes, which causes damage to the area. So this exposure time is about 2 to 8 hours. And an example of this are rare adverse vaccine reactions. You can imagine if you're injected with a vaccine, the vaccine's purpose is to elicit an immune reaction. So sometimes these immune reactions lead to these immune complex reactions occurring. In the figure to the right, you can see that you have the antibodies, which are the little Y's, forming with the antigens, and the complex is created, and then below you see that it recruits neutrophils, and the neutrophils cause damage to the area. Okay, so the last type are type 4 delayed cell-mediated reactions. So the key here is they're cell-mediated reactions, and they're delayed. So these ones are caused by T cell activity. They take a while to occur, sometimes as long as more than 24 hours after exposure, although it can happen quicker than that. Initially, sensitization results when primary contact with the antigen causes the production of memory cells. And then upon the second contact is when the memory cells are activated. T cells, which release cytokines and activate phagocytosis, come to the area. So to the right, we can see this delayed cell-mediated reaction with poison ivy. So poison ivy has a chemical in it called a urushiol. And when that urushiol gets into your system or makes contact with your skin, it can change proteins in the cells. So here we have skin proteins that change, and when they change their structure, they're no longer recognized by the immune system as being from the body itself. So then the T cell has an immune response to those altered proteins. So that's why it's called a delayed cell-mediated reaction. So let's do a little bit of a review. Now, this table is an excellent table for you to cut out and study or review as often as you'd like. We have the 4 different types of reactions. We have how long it takes for the clinical symptoms or signs to occur. We have the key characteristics, and then we have some examples. So type 1 are anaphylactic reactions. They occur very quickly, less than 30 minutes. They involve mast cells or base cells. The IgEs on them basically react with an antigen, which again is called an allergen, and this causes histamine to be produced. This can lead to anaphylactic shock from a drop in blood pressure. This can occur from drug injections and insect venom. They're common allergenic responses such as hay fever and asthma. Type 2 are the cytotoxic reactions. They take about 5 to 12 hours. And the key word there is cytotoxic. So we're looking for some cells that are involved with this. And of course, we have IgM and IgG antibodies that target a cell. And then when combined with action of complement, those target cells are destroyed. So a great example of this are transfusion reactions or Rh incompatibility. Again, if you are a type A, for example, and you receive a type B donor's cells, those red blood cells have that carbohydrate on the outside that is different than yours, and so you'll develop anti-B antibodies that will attack that carbohydrate on the outside of their red blood cells. Type 3 are the immune complex reactions. Again, they don't take very long, about 3 to 8 hours. Not as fast as a type 1 anaphylactic. And these are when you form those complexes, hint immune complex reaction. And so antibodies may bind to an antigen, such as when you get a vaccination, for example. They form complexes, and then they recruit cells that lead to more inflammation and damage of the area. Lastly, we have the type 4, the delayed cell-mediated or delayed hypersensitivity reactions. They take the longest, typically 24 to 48 hours. They involve CTLs, which are just the cytotoxic T lymphocytes. And because this is cell-mediated, it requires an exposure first and an immunogenic reaction first. The example we gave was a poison ivy rash from the urushiol, which changes your proteins that your body then finds to be foreign, and that elicits that cytotoxic T lymphocyte. Other examples of this are transplanted tissues, certain chronic diseases such as tuberculosis, etc. You don't have to worry about knowing the difference in exposure time for type 2 and type 3. They're really close to each other. They're about middle of the road. You should be able to distinguish if it's less than 30 minutes, that's definitely type 1, and it can't be any of the other types. And you should know that, okay, if it's less than 24 hours, but it takes a while, then that's going to be either type 2 or type 3. And type 4 will take usually much more than a day, 24 to 48 hours.
Checkpoint 1. Are hypersensitivities caused by the innate or adaptive immune system?
So moving right along, that covers everything you need to know about hypersensitivities. Let's cover autoimmune diseases. So auto means self, so self-immune diseases are immune diseases where the immune system recognizes self-antigens as being foreign. So that means a loss of self-tolerance. Autoimmune reactions may be cytotoxic, they might be immune complex, and they might also be cell-mediated. So they have names that signify that their functions are very similar to hypersensitivities, only it's to the self. So let's start with cytotoxic. An example of this is multiple sclerosis, where you have autoantibodies and T cells and macrophages attack the myelin sheath of the nerves, and this causes problems with impulse conduction. Immune complexes, it's very similar as we discussed with transfusions and Rh factors. So in this example, we have rheumatoid arthritis, and what happens is you have so much inflammation in your joints that you can have immune complexes form. So IgMs and IgEs or IgGs, complexes develop in the joints, and they're deposited there, and that recruits other cells and leads to further chronic inflammation. And lastly, cell-mediated. And again, we have T cells doing their little thing here. An example is insulin-dependent diabetes mellitus. So we have T cells that actually destroy the insulin-producing cells in the pancreas. So T cells have to be trained properly in order not to attack self-antigens. Most are not well trained. Some have to be destroyed by T regulatory cells, but sometimes things go wrong and autoimmune diseases result. Okay, so now let's talk about transplant rejection. So human cells display a histocompatibility antigen that are encoded by a set of genes forming what is called the human leukocyte antigen complex, or HLA complex. These HLA complexes need to match between a donor and recipient in an organ or tissue transplant. So medical professionals go through HLA typing, which is a process for identifying and comparing HLAs on cells belonging to the donor and recipient. This, of course, is a big deal because if there is not a match, then those HLAs from the donor will not be recognized by the recipient's T cells as being self, and so they'll be targeted and destroyed. However, we should discuss regions called privileged sites and tissues. Privileged sites are areas on the body that are rarely exhibit transplant rejection. Examples of this are the cornea and the brain. And again, privileged tissues are tissues that do not stimulate transplant rejection. Examples of these are human heart valves with pig heart valves. It is thought that privileged sites exist because the immune system has a minimal presence in these areas of the body. Let's talk a little bit about immunosuppression with transplants. The first successful transplants were enabled by the discovery of cyclosporin. So after a transplant, the cell-mediated immunity of the recipient is deliberately suppressed to make transplant rejection less likely. It is done with cyclosporin, which inhibits the activity of leukocytes, especially T cells. Sirolimus is also used in cases of chronic or hyperacute transplant rejection, and this inhibits both the cell-mediated and the humoral immune system. However, scientists are working on using stem cells, which can differentiate into different tissues and organs, to grow new organs outside of the body, which is becoming more popular. It has been done with urinary bladders, and it has been done with lungs and kidneys in pigs. Someday we'll probably be there. Here's an example where scientists used it to save a boy's life. He had junctional epidermolysis bullosa, JEB, which is a severe and lethal genetic disease, which has mutations in skin cells. The scientists actually took a sample of the child's skin cells and used a adeno-associated virus, which is just a vector used to deliver genetic information, to deliver the correct mutation on it, and allowed that stem cell to grow into sheets of scales that were used and transplanted back to his body, and now he is completely cured. Okay, let's get back to our list of types of immune system disorders. The last type we'll discuss are immunodeficiencies. So immunodeficiencies are like all the way on the other side of the spectrum. They are actually diseases that are characterized by the lack of a sufficient immune response. They can be congenital or acquired. Congenital immunodeficiencies are present at birth, and they are the consequence of genetic defects such as the DeGeorge syndrome, which is characterized by a lack of a thymus gland, so there's no cell-mediated immunity. That's because T cells mature in the thymus. Acquired immunodeficiencies can develop as a result of drugs, cancers, or infectious agents. So the example here is Hodgkin's lymphoma. Hodgkin's lymphoma lowers the cell-mediated response. Lymphoma is a type of cancer that originates within a specific type of white blood cell called lymphocyte. And here's a table showing selected immunodeficiencies. It's nice to review them a little bit, but they're not going to be on the test. Except for acquired immunodeficiency syndrome, or AIDS. In this case, the virus ends up destroying the CD4 T cells, and this allows cancer and bacterial, viral, fungal, and protozoan diseases caused by HIV infection. So first, let's do a little etiology of acquired immunodeficiency syndrome. It's important to identify the difference between the syndrome or disease and the agent that causes it. So here, AIDS is a disease that sometimes develops after an infection with human immunodeficiency virus, HIV. Note that the virus has a V in its name. You need to be aware of certain traits of HIV. First, HIV is a retrovirus, so therefore it contains RNA as its genome. It has 2 identical plus strands or sense strands of RNA.
Checkpoint 2. What type of virus is HIV?
So at stage 2, viral replication occurs at a low rate compared to the initial stage of the infection. The T cells start to decline, and they decline at a pretty steady rate. And this, in part, leads to early signs and symptoms that the immune system is falling apart. So symptoms might be yeast infections, or diarrhea that's persistent, or a fever that's persistent. And this can last as long as 10 years or more. The natural progression of the disease leads to stage 3. And this is where we have development of clinical aids. The T cell count drops below 200 cells per microliter, from 500 to 1500 microliters. And symptoms begin appearing, including yeast infections of the respiratory system. They might easily contract tuberculosis or toxoplasmosis of the brain. And before this point, a person is considered merely HIV positive. At stage 3 is when they begin to have AIDS. So this virus infects roughly 36 million people worldwide. About 70% of cases are in Africa, and about 13% of people infected with HIV are in the US and are not aware that they are infected. Lastly, 1/3 of cases are in Europe and Southeast Asia, and they're from drug use.
Checkpoint 3. Reverse transcriptase exhibits a higher mutation rate than other DNA synthesizing enzymes. Explain why this makes HIV difficult to treat.
So let's talk about treatment for HIV. Because they are a retrovirus, they have a very fast mutation rate, a very high mutation rate. And that prevents scientists from developing effective vaccines because the targets are always changing. There are different drug classes that are used to treat HIV, and they include reverse transcriptase inhibitors, integrase inhibitors, and different proteases that the virus uses, different protease inhibitors. And a possible future treatment may involve gene therapy related to the CCR5 delta 32 mutation. This mutation is found in certain populations of humans around the globe that leads to HIV resistance.
Checkpoint 4. Elvidagravir is a drug that prevents the formation of an HIV provirus genome into the T cell genome. How would this drug be classified?
So a scientist tried this in China, and while he did introduce a mutation, the mutation was not in the exact spot that it's seen in individuals who have resistance to HIV. It was a little upstream or downstream within that same gene. So people are upset for many reasons. One is we don't know that it was necessary for the fetuses to go through such a risky procedure, that he did not get permission from the parents. He did it without their knowledge. We don't know the consequences of having a mutation elsewhere in that loci of the genome. But nonetheless, this is the first open sighting of a scientist who has edited human babies. So here is another research article entitled Prevention of Vaginal SHIV Transmission in Macaques by a Living Recombinant Lactobacillus. And in this study, they actually introduced a probiotic, a lactobacillus called Lactobacillus jensenii, which secreted antibodies against HIV. So they genetically engineered it to produce these antibodies that were specific for HIV. And as a consequence, they reduced SHIV, which is simian HIV, which is just monkey HIV basically. They were able to reduce the rate of transmission by 63%. And this final research article is where this is the second case of reportedly curing HIV in a patient. And the way that they did this is it was actually done with a bone marrow transplant in the individual. But the donor actually had that CCR5 mutation present. So the bone marrow that was received had the mutation which confers resistance to HIV. And that's the end of chapter 19. Thank you so much for being here, and we'll see you next time.